Watch one cell in a dish break apart while the cells right next to it keep growing. Nothing poisoned it, nothing reached in and cut it from the outside. The only thing different about this cell is one specific RNA message it was carrying. And a repurposed CRISPR protein read that message as an order to shred the cell's own DNA from the inside. Here is the plain version of what you are looking at. This is a CRISPR protein called Cas12a2. Like other CRISPR tools, it uses a short guide to hunt for a matching sequence. But most CRISPR proteins you have heard of make one careful cut to edit a gene. Cas12a2 does something blunter. When its guide finds the target, the protein switches into an attack mode and starts chewing through DNA broadly, all over the cell, enough to wreck the genome and kill the cell that held the target.
That difference is the whole point. It means you can pick a cell by what it is saying, not just by what its DNA spells out. A guide aimed at an RNA that only appears in an infected or cancerous cell becomes a switch that removes that cell and leaves its healthy neighbors alone. The work comes from a team led by Ryan Jackson's lab at Utah State University, with collaborators at the University of Utah, published in Nature in May 2026, and a follow-up from Jennifer Doudna's group in June that pushed the idea straight at cancer.
To see why this is strange, line Cas12a2 up against its famous cousins. Cas9, the gene-editing workhorse, makes one precise double-strand cut at a spot you choose, and the cell usually stitches it back together. A protein called Cas13 goes after RNA instead of DNA and can chew up RNA once it is activated. Cas12a2 sits in an odd place between them. It is triggered by finding an RNA, like Cas13, but then it turns around and attacks DNA, savagely, like a saw with the guard removed.
This behavior did not start as a medical idea. In bacteria, Cas12a2 is a last resort defense. When a virus infects the cell and its RNA shows up, Cas12a2 fires and destroys the cell's own DNA, killing the infected bacterium before the virus can copy itself and spread to the rest of the colony. It is a self-sacrifice system. One cell dies, so the population survives. The May paper's move was to take that suicide switch out of bacteria and get it working inside yeast and human cells.
They built up the evidence carefully. In Baker's yeast, they wired a guide to a chosen transcript and showed that turning Cas12a2 on wiped out the cells carrying that RNA, while control cells came through fine. Then they moved into human cells and delivered the system two ways: as ready-made protein and guide packages, and as the genetic instructions wrapped in lipid nanoparticles, the same kind of fatty bubble used in mRNA vaccines. Aim the guide at a marker transcript, and the cells expressing that transcript dropped out of the dish.
Two comparisons in that work are worth pausing on. When they used a Cas13 protein against the very same target RNA, it only thinned the cells out modestly. RNA cutting alone wasn't that lethal. Cas12a2's DNA shredding hit far harder. And to prove the damage was real DNA damage, they lit up the cell's own repair alarm, protein markers that pile up at broken DNA ends. Those alarm sites bloomed across treated cells at levels the authors compared to harsh chemotherapy agents, but only when the target transcript was present. Then, they pointed it at real disease targets. They aimed the guide at the RNA of HPV, the virus behind most cervical and many throat cancers, going after the viral E6 and E7 transcripts that infected cells keep switched on. Cells carrying HPV were depleted. Cells without it were spared. They even treated a mouse carrying a human HPV-positive tumor by injecting the lipid nanoparticle system directly into the tumor. They also went after a cancer mutation in a gene called KRAS, targeting the mutant transcript and sparing cells with the normal version. The June follow-up sharpened the aim at some of the hardest targets in cancer. Roughly half of all tumors carry a mutation in a tumor suppressor gene called TP53. When that gene works, it helps damaged cells shut themselves down. When it is broken, cells that should die keep growing. The problem is that a broken, missing function protein gives drug designers almost nothing to grab onto. There is no neat pocket to jam a small molecule into. These are the targets people call undruggable.
Cas12a2 sidesteps that problem by ignoring the protein entirely. The mutation still shows up in the cell's RNA, the transcript copied from the broken gene. That transcript is a sequence, and a sequence is exactly what a CRISPR guide can be built to recognize. So Doudna, Jingquan Zheng, and their colleagues used the mutant TP53 message as the trigger and let activated Cas12a2 shred the chromatin of cells that carried it. In mice, delivering the system by lipid-nanoparticle shrank tumors and slowed their growth. Step back, and the shift is easy to state. Gene editing changes what a cell's DNA says. This tool reads what a cell is currently expressing, and if it sees the wrong signature, eliminates the whole cell. That is a different kind of handle on disease, useful exactly where the problem is a cell state rather than a fixable typo, a virus hiding in some cells, a cancer defined by a mutation you cannot drug, maybe someday a rogue immune cell. Now, the honest limits, because they are large. This is still work in cell cultures and mice, not a treatment anyone is receiving. Getting the system into the right cells throughout a body, not just a tumor you can reach with a needle, is unsolved. The team reported little off-target firing in their tests, but that is a promising early result, not proof that it stays quiet across every tissue and every guide. And the whole approach only works if the target RNA is genuinely unique to the cells you want gone. If healthy cells whisper the same transcript, or a tumor is a patchwork where only some cells carry the mutation, the selectivity that makes this exciting starts to fray.
Still, the core idea is a genuinely new lever. For years, CRISPR meant rewriting a line of the genome. This turns the same molecular machinery into a sensor with a kill switch. Find a specific RNA and take out the cell that made it. Whether it becomes medicine depends on delivery and safety work that will take years, but the demonstration is on the table. A protein that decides which cells live by listening to what they are saying.